So, Sarah, nice to meet you digitally. I don't think we've met in person before.
Yeah.
Um, Very nice talk, very interesting. I don't know a ton about optogenetics, and so I don't know how naive my question is, but it might relate to the silent engram
Okay.
idea. So I
Yeah.
was thinking specifically about the false memory results.
Yeah.
And if I understand things correctly, the order of operations in the experimental design goes like this. You first form the engram in chamber 1.
Mm-hmm.
Step 2, you get doxycycline to prevent the formation of a new engram subsequently. Then you're moved to a second chamber, and the original engram is then optogenetically or optically
Yeah.
activated. And at the same time, foot shocks are administered. Um, and those foot shocks are described in your paper, and I think in the Tanigawa or Redondo papers, as sort of giving the engram a negative valence or
Yeah.
something like that.
Yeah.
Right, so it's the same old engram, but now it's acquired this, to use philosophers speak, accidental property of a negative valence, right?
Mm-hmm.
And then the mouse is put back into the original chamber, demonstrates the fear response, and that's supposed to show, well, at least according to your interpretation, that's the evidence, like the demonstration of the original fear response shows that it's it's not a problem of ekphory, it's a problem of, it's to do with the engram, not the ekphory.
Yeah.
Right?
Yeah.
Okay, so that's the setup. That's clear. And now here's my question. Maybe this is just a cheap trick, but in step 2, when we said you go to the second chamber and the original engram is reactivated,
Yeah.
why can't, like, what is it in neuroscience that prevents me from saying maybe it's not the original engram that's reactivated. Maybe it's, here's the alternative, maybe it's a new engram that's being formed using the same neural resources. Right, so I'm being philosophical, I'm pushing you on the definition of engram, right?
Yeah. I think
But if you say that, then I think the result is that when the mouse returns to the mouse returns to the original chamber, it is at least possible that it's a problem of retrieval. It's a problem of ekphory, not a problem of the engram itself.
Yeah, no, thanks. I think it's a very good question. It's a question I've started to have a version of myself. I mean, there's one way of blocking at least some of the skepticism, which if I'd gone through the series of their papers in more detail, I think would calm some of the concerns, is that they have a set of original papers where they really try to show you can, optically tag an engram and then turn it back on and turn it back off. And so they do a set of ones where they just track that engram with light over time and don't do any manipulation, where you do see, so imagine in that initial context where you're forming the neuron that's going to express the opsin, you are being conditioned in one respect or another. I can take you to lots of different contexts and turn that on and get you to display the behavior learned in that particular case. So
okay, right.
So they do establish in previous papers that you can form and then reactivate this engram prior to manipulating it. But I think even with that said, there are these real worries about, this is a point the more I get into the weeds of this and some related stuff, the more I'm convinced by, the more my philosopher hat is showing, I guess, in the ways that you're suggesting, which is that we have to have pretty serious ideas about what it takes to be the engram, and in particular about what it is to stay the same engram over time. That's
my main
about the identity conditions of the engram, and it turns out not all scientists have the same view of that, right? So, and different people will say, well, it's the same cells, but I'm getting different behavior. Some then say, so that means engram, so that means new engram.
Yeah.
And you say, like, no, that means distorted engram. And that's really reflecting these differences of commitment. So I'm now moving more towards thinking that, like, this isn't going to be the tool by which we solve things, but it is going to be the tool by which we really get to press one another on what those commitments are. But that's the nature of that commitment is still going to come from us.
Yeah. The identity conditions of the engram sounds terribly pompous, but I suppose that is, in fact, what I'm worrying about.
Yeah, I've been saying it to you, so it's okay.
Thank you.
Thank you, Charles. Now, Ernesto, have you fixed your microphone? Let's check it.
Okay, what about now?
Yeah, now it goes.
Okay, perfect. Then first, let me to say to Sarah the presentation. I am a neuroscientist myself, and the presentation was really, really very good. I mean, I wish maybe the teachers in neuroscience have the ability you have. Then that was a very good point. I would like to, however, to make some observation about what you have said. And well, let me to start. First, I will would like to point that the fear contest paradigm is not just fear of the contest, but of the pain associated with it. Electrical induced pain is not the most natural form of inducing fear, and I think it's a quite extreme form because the animal has no chance to escape. Even though you cannot constrain the animal ability to deal with a threat. I mean, here I will try to remember a little bit Kurt Goldstein, when he was used to say that the reaction of the animals are expression of the organism's attempts to deal with certain demands on the environment. Therefore, the behaviors are attempted solution from the demands. Here there is not so much flexible outcomes for the animal, and that might impose itself in some form of brain activity. Yeah. For example, when the demands are about broad, complex form of learning, as schemas, for instance, Richard Moore's team in 2006 showed that in complex form of learning, not in the cortex, which was actually surgically removed. In other words, a multiple track or single track episodic events are coded also in the cortex, not only in the hippocampus when the complex memories process occur. Then a second point, I go into molecular aspect of that biology is that and the behavioral part also how it relates to that is that it is not necessary to induce pain in such severe way to learn a spatial test, but it is required to get optogenetic experiments to work. Let me explain this part because I think it's methodologically quite important. Okay. The option protein activated by light requires a promoter, CFOS. Yeah. CFOS is an immediate early gene. They are activated during the different stages of activity of cells. And there are other immediate early genes that take more or less time and depend on more or less of the state of activation of the cells. Right. Sarah, are you there?
Yeah, I'm here.
Okay, good. Now, what is interesting is that the important thing to note is that all the experiments, in order to induce a mental paradigm, context, fear conditioning, electrical shock, quite natural. Well, this is in part because you need a very high activity in order to turn on the activity of some group of neurons. In fact, you only see what seems to be excitatory in the nerves. And it is clear that this population is necessary to trigger, to a certain extent, a response. But the experiment does not reveal the full complexity of for instance, because it doesn't show the inhibitory neurons. Surely they are playing a role. No neuroscientists think the inhibitory neurons do not play a role in the engram formation, but you only see excitatory neurons in most of the studies until now. It is possible then they're using a broad diversity of immediate early genes. Other type of interneurons we will be able to see them, and then to have a very picture what the engram is at least in relation to the fear context. Now I come to something that is more important behaviorally, because you see in the first paper. You can read very well the compare with the experimental group, the light-induced freezing levels after improving times and stimulating both sides of the hippocampus reach maximum 35%. This is quite important. With those typical but you compare with those typical reported to conditional context in animals, it's 60%. This is quite a difference. Then the animals are not being recovered fully, like in the control conditions. It's only until 35%. Then the memory recovery is not perfect. Let's put it that way, not complete. Then there are problems with the optogenetic method cells, because even if it allows the target activation of the neurons, it activates them all at the same time. Okay? Yeah. Then you are not really able to capture the space and time dimensions they might really occur when the animal actually normally is actually, the engram is being activated. Then that cannot be, let's say, that cannot be done with optogenetics because that is not that technique doesn't allow you to do that. Now, then just to conclude that's to put in now let me put something a question. Even if the activation of this partial engram yields a consistent behavior outcome, the problem is the memory content under natural conditions uploading the same memory experience. In other words, is the memory experience of the mouse the same, does the mouse, has the experience of that event in the same way artificially the natural way. Maybe that is what would be interesting to look at the humans. My guess is there will be not the same thing. Anyway, now I will bring it to a last point. I don't know if I'm using so much of your time, but let me to put on a last time more related directly with my research. The interpretation of the experiment relying in a measure of how the tacit implicit form of memory is retrieved with accuracy. But this creates the assumption that an organism's adaptive capabilities are purely dependent on some sort of objective capabilities to capture external information. But inner states, emotional states at the moment of the event could also configure differently what is remembered and what is retrieved. Now the problem is that in the state where the fear conditioning has Tonegawa group has carried out these experiments, there is little flexibility for the animals to have other emotional states. Okay.
Yeah.
For instance, I just want to put a little bit matter of proud a little bit of work we carried out the last year in natural neuroscience. We for instance showed that ventral hippocampus produce a form of theta 2 activity associated with anxiety. Okay. Now the interesting Tonegawa in 2016 showed that a form of social behavior memory associated with the ventral hippocampus. He said that there is engrams associated with social memory in the ventral hippocampus. Well, the ventral hippocampus also have cells, if, in this case interneuron. They control the activity of excitatory neurons. They also are related with anxiety-related behaviors. Then you have, from that perspective, you have in the same regions groups of neurons. They are related with engrams, but also related with emotional-related behaviors. This brings me to the conclusion then neural components in that brain area might be reused for different purposes. You see? Then there is not and surely then some of these I wonder, are these engrams overlapping with the anxiety related circuits or not, or this kind of thing? What does it say about memory? What is it about emotion in distinction with memory process and cognitive process? Now, I don't know if I have time. I have more time, but I have okay.
We have still many questions, so we will check if there is more time. We can do that also.
Okay, no problem. Okay.
Also things to answer for Sarah, so.
Yeah.
This one.
Ernest, this is great. Thank you for all of that. I'm gonna just assume I can email you for some more details about these things. So I feel like I'm still in the midst of learning about what some of the limitations are to get optogenetics to work. The comments about CFOS in particular, I was not fully aware of, and I think that's. I had questions about what they were using for the positive stimuli and why, and maybe what you're revealing to me is explaining part of what they've got to do to get the level of expression that they need. So, yeah, it's a lot of things to respond to. I think that's certainly going to limit what it is we can ultimately infer from the kinds of results that are going to be possible, and the amount to which we'll be able to use optogenetics to test all of the questions that we have about memory. So bracketing the things you said at the end about sort of context and neural reuse, because I think that gets back to a lot of the earlier, it feels gross to keep calling these identity condition questions, but sort of questions about how we're going to track these cells, and whether we think the anxiety states and various other networks are part of that same, whether they're going to be part of the engram or not, or whether they're separate processes. I think those are theoretical commitments on our part, or on the part of particular paradigms for researching, for doing this kind of research. They're not going to be given to us by what we see. So I think it's at least worth exploring, different ways of theorizing about these things. But it could be, so we might not be able to test everything with optogenetics, but we might, the fact that we can manipulate and move around some memories is at least, in some cases what we need is sort of proof of concept that it can be done in particular cases. So maybe we don't know that all memories will work this way, or that if the animal had more options that they would always do this sort of thing. But we have at least something in the way of ruling out some alternative hypotheses, perhaps, about the mechanism involved. But I want to talk to you more about some of these limitations, and I'm grateful to you for bringing this to my attention. So, thanks.
All right. So thank you, Sarah, and thank you also for the previous, I guess, commentators. So Ernesto went on to one point that I was going to make having to do with the extent to which the genetics actually is really altering the whole circuitry of what one might think it is the engram. But the thought that I had while he was talking is, well, you might not even need optogenetics to make the claim of something like artificial equity, because for instance, from work by Wilfred Penfield in the 40s, that if you excite the cortex of an individual during neurosurgery, they can't help but remember certain content. I think you might remember that. So Penfield will open the skull of the individual in the surgical table, and then starts probing here and there. And what he started documenting, and in fact part of this evoked sort of potentials, as he called them, during the surgery, was what allowed him to come up with a homuncular maps cortex. But they were, as it were, completely artificial exploric moments. That's not how we go about remembering stuff. We don't go about what I need to go, go grocery shopping this evening. Let me just poke my head in the grocery list area to see what I have to remember. So it seemed to me that even if optogenetics would not fully reinstate whatever circuitry is supposed to be reinstated, which I think Ernesto just clearly articulated, there's still other examples that clearly sort of support your notion of artificial equity. So that being said, I have two very sort of general questions. Why is it, I guess the first question is, why isn't your argument, or why isn't your paper an argument for eliminating the distinction between equity and engram?
Yeah. Is that just the question?
The reason for that is Semon had a view of an engram. In fact, he was interested, and I love the way in which you put it in terms of the Mendel of memory, because he was interested in the passing of engrams through evolution. So he was interested in one single brain. He was interested in information that passes from individual to individual, and he had a relatively stable notion of engram. Whereas equity is what gives it kind of like the activity to it. But it seems to me that nowadays, whether you're a constructivist about memory or not, no one believes in an engram as stable as Semon originally believed it was. And I wonder whether by just changing that foundational notion or foundational property of an engram, thereby you're not sort of forced to reconsider whether this conceptual distinction is actually a distinction that is sponsored by the nerve system.
Yeah, that's a great question. I'm glad this is going to be recorded so I can have the way that you put that there. So I don't know that ultimately I want to go as far as you in completely getting rid of that distinction, but I certainly don't want to celebrate the reification of it in the way that I did when I first wrote this paper. So I think that, so I'm thinking back more and actually reading the original Zeman more, which is not easy and strange. And in his talk about the expiry, in particular, trying to make connections between the engrams in one person and engrams generationally. His ways of talking about the expiry don't distinguish much. This is also getting back to a point that Ernesto was making between the sort of internal expiry, sort of the overall expectation levels of the system, and the environment and the context and things in which they're occurring. So the expiry is doing way too much. And so it might not be the fault of everyone else for not paying enough attention to what he had to say about it. It might be that context wasn't clear enough to begin with. And so there might be more to do to say how that distinction might change. I don't know that I'm ready to get rid of the entirety of it. I agree that there's not going to be a stable and persistent thing, but there might be some notion of stability, or you might want one, or at least one way of distinguishing sort of the thing that's in there from the processes influencing it. Maybe I'm not sure I'm ready to give that up yet. But you'll probably talk me into it at some point.
Yeah. And I guess that one last point is somewhat related is, you know that I have thought about memory traces in the philosophical context quite a bit as well.
Yeah.
And one of the things that is kind of like interesting about the simulation view, and this is something that's different between Kirk's view and my view, is that I don't want to get rid of the notion of memory trace at all. And it is kind of weird to be a constructivist and at the same time like hold the view that you require something like memory traces. But I wonder if, so the view that I have been pushing lately is the idea that the notion of memory trace could be either something that is categorical, that is to say something that exists and it retains content all throughout and is probably wrong, or you can think of memory traces as a dispositional property of a neural network to reenact the state it was in during encoding at the time of retrieval. If it is a dispositional property, then I think that the sort of expiry and engram distinction kind of collapses a little bit. So I mean, if the distinction kind of collapses just because what a memory trace is the disposition that gets actualized given the right cue or what have you, then don't you think that notion of memory trace might be better to challenge, to sort of face the challenges that you pose?
Yeah, absolutely. So I think we're broadly in the same ballpark. I think a lot of the work is, it feels to me like you've been someone committed to being a sort of constructivist simulationist who wants memory traces to somehow work out. And I'm somebody who wants there to be memory traces, but wants to acknowledge everything that the constructivist and simulationist is saying. So the answer is somewhere probably in the middle there. I like the move toward dispositional way of thinking about things. I think a lot is going to hinge on exactly what the dispositional account is underneath that. And some accounts of dispositions are going to have room for a distinction between sort of the possession of the capacity and its manifestation, and other views that distinction isn't going to make sense. And so I think whichever one turns out to be right, assuming this is the right way to go, is probably going to sort of, tell us how to make those kinds of commitments. At least, that's what I'm thinking.
Okay, yeah, I think that you're right. It's going to depend a whole lot on what you mean by disposition, but also I think that your work once again shows that categorical views of
Yeah. I'm gonna beat it into the ground. That's
Right.
Is that noise on your side?
I don't think so.
I don't think I was making any noise.
Okay, apparently it's not your fault. Sorry. Okay, so welcome. Time for your question, by the way.
Thanks. Well, now I will make some noise. Is that maybe feedback? Double speak? Anyway, you guys can hear me.
It's feedback.
So, thanks, Sarah, for the awesome talk. I really enjoyed it. I think one thing I might want to do is maybe generalize some of the questions that have been raised so far. So I really, found this science very fascinating. One maybe issue I had, so the positive thing about optogenetics is that it's so fine-grained spatially and temporally, right? That's the positive thing. But I guess it was a little uncomfortable about using that to test sort of psychological constructs that are much, much less fine-grained. So you've got this very broad distinction between the n-gram and the ectory. And if you're to interpret that literally, then that implies that there's this sort of real distinction between these processes. And then one way to maybe interpret some of the questions people have had is that, like, it's unclear whether the actual manipulation speaks at the right level to really tease apart those different commitments of the much broader psychological distinction. And my suspicion is that when you actually get into the fine-grained manipulations, they're not going to precisely fall out in a way that captures the broader psychological distinctions. I think this hits on some points that you and I have talked about over email. So here's just one example. I don't know if I've mentioned this paper to you before, the Place and Eichenbaum paper from 2014. It's a Nature Communications paper. And what they do in the paper is they use Granger causality over different stages of a memory task. And what they show is that as a mouse initially enters a context, you get driving of frontal responses by hippocampal responses. But as soon as the mouse starts to behave in that context to perform a particular thing that they're supposed to do in that context, the directionality shifts, right? So now you've got prefrontal activity leading hippocampal activity. So, I mean, one of the things you might say is that when you get to this fine-grained level of interaction, either they're both involved in encoding memory and retrieval, or that they're flipping those roles at some stage, right? So one way to describe this is the hippocampus is doing initial recognition of the context that's driving a recognition of the context in the prefrontal, but then what the prefrontal is doing is driving a recognition of the required rule in the hippocampus in the latter stage of that. So I mean, which one of these is like storing the memory about the context, and which one's doing the retrieval? I don't know. It just seems to me that there's not a very obvious answer in this question. And you might do opto, and you might find some differences between interrupting these different processes at different stages of the task. There's a great paper by Gord et al. in a working memory task where they interrupt a bunch of different areas at different stages of the task and look for exactly which areas are necessary at which stage. So you could do that, and you could get these really nice, necessity claims like this process is necessary for. But that seems to me to fall short of establishing like this is where the content is stored, And it seems like the n-gram view is kind of committed to that.
Yeah, that's a great way of pushing that point. I owe you an email, and now I owe you a bunch more. But I think that's absolutely right. And there's a way in which, part of the fascination for these papers initially was really seeing this group in particular really go for it and trying to make the connections to the cognitive stuff. I've been interested all along in sort of seeing how much of that promissory note they can cash in on, or whatever metaphor I want to continue with. But I think as the work has gone forward, and a lot of the stuff on the silent n-grams that they've been doing more recently that I've been paying attention to is really starting to complicate the notion of what the n-gram is or what view it might be supporting. So you've got this idea like all the cells are involved at different times, and often genetically you can make any of them do what you want, but it's not clear that that's how they're being used in the system at any particular time or what kind of information they're holding. And I think it's especially complicated because the memories we're talking about here, the content we're talking about here is usually behavioral and spatially constrained. So it's exciting to see the translations to animal models in certain respects, but insofar as many of our questions aren't just cognitive questions, but are conceptual, sort of about the structure of the representation and how that changes over time, then lately I've been feeling like we've hit as far as we can go with scaling this stuff up because of that. And I don't know if I'm going to stay in that grumpy space forever, but at the moment, just feeling like I'm feeling the disconnect more than I wanted to. But that's precisely my worry, is that if the memory is just, was I in context A before and was it fun or painful, that doesn't have enough content to it in the right sort of way, or is the food in the right corner, that doesn't have the right kind of content to be manipulable in the ways that we care about for theories of memory and other things.
Mm-hmm. Well, can I follow up really quickly? I mean, so I mean, one way I think to be optimistic about it is, so I agree with everything you said, and I think it's like really interesting to carry forward with this and see where this goes. One way to maybe be slightly more optimistic about it is to say, this is a way of really kind of understanding the limits of the opto method, right? So I'm really interested in multiplexing, and it turns out in this Gore et al. Paper that I'll send you if you want, they look at a comparison of P1 frontal motor and posterior parietal, and it turns out that all of them are like multiplexing 6 different variables that are relevant to the task. I forget what all the variables were, but it's stimulus, rule, response, and a couple other things, maybe past response. But then, in a sense that under the fact that they're all responding to those variables underdetermines what's, the crucial role they're playing with those variables. And they kind of use the opto as a way of testing out, what the functional contribution of integrating those variables in the particular way. So anyway, I mean, I think there's one way to think about how to relate the physiology, like more standard behavioral physiology, to optogenetics here, and that sounds positive to me. I don't know, it's one of the most hopeful methods I've discovered recently. So
yeah, well, good. That's I could use a little hope. So thanks.
Okay, thank you, Sarah. I would like to know your conceptual appreciation about this. I mean, for me, I have a problem with the use of the word falsehood to refer to experiments when optogenetics is used to trick the animal and make it respond as if remembering wrongly. I have the impression that it brings me some misinterpretation. Taking into account that I have been working in memory. My work has been in memory anxiety in Veterano Campus, okay? Now, what is investigated from my perspective is the memory accuracy in a very restricted context, but not the subject of state of falsehood about a past experience. I mean, I have a friend of mine, Samuel C1, he is a neuroscientist here in Uppsala University, who told me once, I wonder if an entity remembering everything with accuracy could be adaptative in any sense. Then the question, putting in other terms, I almost see Gram not as an objective representation of the world, but instead has an interiority of the subject that grants a meaning to it. I mean, it doesn't mean that Gram's local, or in some cases distributed across the brain, are not fundamental aspects of memory. But it's supposed that the concept of memory is not just about storing information reliably. But I wonder, if I am a biologist, if we should ask to what extent memory, does they look like memory failures related with some sort of cognitive adaptability? My problem fundamentally with the views of memory that Tonegawa team work out is that the concept seems that all form of memory is a sort of pure dysfunctional aspect of the organism. And I have doubts about that, seriously. What is your opinion about that?
Yeah, so in some ways this question is better for Felipe than me. I think he's been one of the leading people, especially in philosophy, of thinking about more nuanced ways of thinking about what the function and purpose of memory might be. So the idea of, maybe I've been bashing on the view of memory as this kind of perfect objective picture of what the world is like at any particular time. I think insofar as anyone has ever actually held that view, and I'm not sure many people defend and articulate that view as much as people put that out there as an alternative view. But that view strikes us as wrong for a lot of reasons about what memory is doing, and that whatever notion of accuracy is going to be involved is going to have to be weaker and more contextual and more sensitive to subjective variables. I think that's absolutely right. And so a lot of the work in both more psychology and philosophy of memory has been about rethinking what accuracy of memory, what the right notion of that, if we even need one, should be, what the overall function of memory might be. And some people are happy to be relatively extreme on that view and say, it only matters that it's sort of what might have been. There's not any strong accuracy constraint. I don't want to go quite that far, but I agree that the point of if when this talk seminar ended today, if I remembered the number of words that you said rather than the general content of the objections you raised, I'd be missing the point, right? And my failure to remember the number of times you said neuron is not a reflection of my ability to remember the event. So we want to make sure we have a notion of accuracy that's, or at least some kind of constrained by what actually occurred notion that's suiting our purposes. But whatever that turns out to be, it's clearly the case that in humans we do this, right? We we remember events and we blend them together, and even if we don't want to say that the mouse is misremembering or has a false memory, they're at least, you might still think that they're doing something similar to what it is that we're doing. If that's, so regardless of how you want to label or characterize what those things are, it's well documented that we do these sorts of things, and now we've come up with some sort of experimental paradigm which shows the ability to concoct something similar in a much lower organ, in a rodent that we didn't think we could pull off. And that, to me, is perplexing enough to spend time thinking about.
Can I say a thing about that?
Yeah, you're welcome. I was expecting that.
So Sarah is right in that I do believe, and I have believed for a very long time, that that there's a large number of false memories that are not the product of a malfunctioning memory system. That doesn't mean that every false memory is equally on a par with an accurate memory. It's just that accuracy and adaptability are not tracking one another perfectly well, which I think is what Ernesto was suggesting.
Yeah.
However, the memory system does, in an important sense, try to get things right? A memory system that it is 50-50 in getting things right is not going to be really great, and that would be just bad. Like if your rate of false alarming is just the same as your rate of giving you a signal, you don't have a very, very reliable memory system. So one thing that I think is very interesting is in just the research in memory in general is to try to figure out what are the boundary conditions for false memories that are just the product of a well-functioning system, and which are the false memories that go beyond that boundary condition. And then the second point that I was going to make is this is one of the difficulties of the scaling up research in memory with animals to humans, right? Because accuracy is always dependent on the task. So in humans, we have different measures of accuracy. In rodents, we're going to have different measures of accuracy. You don't expect a rodent that has been, subject to the optogenetics treatment to falsely remember, that he was being bullied by another pup.
Yeah.
He was a little baby when the task for accuracy only asks for a fear or sort of response. So we have in other words, the realm of interpretability of what constitutes accurate is so incredibly confined to the particular task at hand that sometimes it's not very clear how are we going to scale accuracy in that task to just truth in general for us. So that would be the other point that I make. I love research on animals, but I am also incredibly also human animals, but I'm also very skeptical of to what extent can these very general terms that we attribute to a phenomenon like memory are generalizable from single tasks that confine the interpretation so much.
Okay, well, I think so that Sarah has a very important point, and I think so. As a biologist, I was very impressed with the Negawa group when they demonstrated or showed, for instance, the protein synthesis was highly associated with the retrieval process, with the gram formation. I do think so. I do think that is from neurobiological point of view is extremely important. I mean, but I don't think so that is a paradigm shift or anything like that. I do think so that it shows something, but that presents another important problem is this: that you have a time dimension that distinguishes between retrieval and gram formation is a time dimension, but it doesn't mean that the space dimension still will not be able to overlap a certain moment later on. Put in this way, imagine that some of the synapses that require protein synthesis for retrieval are in the amygdala. Now you could imagine that synapses in the amygdala that have been recruited for retrieval in the amygdala in the future will be maybe part of an engram as they is formed in the amygdala itself. Then anyone has so far have been able to. I agree that distinction between retrieval and engram is being presented very nicely in relation with this specific paradigm, but it doesn't rule out the possibility that synaptical protein synthesis associated with the retrieval component might not be able to be recruited and form part of an engram later on. What do you think about that? I think there is a possibility that it could be very interesting because it meant that a memory can help you. The retrieval engram associated with a memory can help you to relate with another memory. So somehow.
Yeah, sort of interconnections between them. So I'm not sure if I'm. Yeah.
Because, synapses are, when people talk about synapses, it's not one-to-one. Synapses can come from extremely complex. I mean, you can have, many in very small reduced spaces.
Yeah. Yeah. I think that's a very good question. I'm not sure I have anything all that sophisticated to say about it yet, but I think maybe this is a way of taking on some of Dan's optimism, that much of the work that's been doing recently about the sort of silent engrams, which stems from the work that started on consolidation and the protein synthesis, is work that shows that certain cells aren't ready to help in the retrieval. They have the information or they have a copy of it or something, but until they have a certain, or only when they have a certain amount of dendritic spine density can they actually participate naturally, and they go through stages where they gain and lose that over time. And I think that's really fascinating for getting us to think more thoroughly about what those mechanisms might be. And yeah, I mean, it's interesting to hear your response as the neurobiologist thinking about where that leads you to think, which is different than how I think about them. But